Welcome. This is the video lecture on cells for the very first lecture in this sequence for this particular course. I'm going to attempt to use this particular whiteboard in conjunction with my own personal PowerPoint slides. The purpose is to give you a synopsis of the main salient points of the reading assignments that you need to do prior to coming to lecture. Let's begin by showing you examples of how this works. On the next slide, in every case, I'll have an agenda, and the agenda will list the contents of that video lecture. And most of these will pertain to the pre-lecture activities. So let's go forward and show you how this works. So I will tackle each of these bullet points one at a time, and I'll use, as best I can, my mouse and my tablet to draw on the slide salient information and talk to you over the microphone about concepts that are very important. So let's start with cell theory. Cell theory is basically an attempt by scientists to understand life. And the component of life that's been discussed by the cell theory has to do with the cellular nature of living things. We'll come back to that in a second. The important thing for you is to understand that you must know cell theory. It's a fundamental concept in this course. There are three parts to the cell theory. Students easily remember two of them, but they tend to forget the third one. So what you should do is remember the hardest one out of the three, and then the other two should be able to be deciphered simply by working backwards. I'm a very visual person, and what I like to do what I like to do is use diagrams, either from the reading materials, the textbook, or generate my own diagrams to help me understand information. So in this case, I've come up to the conclusion that a stool with 3 legs is a great way in which to remember cell theory. So here we have a 3-dimensional stool, and it has 3 legs. The first leg can correspond to the first statement, that all living things are made of cells. That's what it says. So all living things are made of cells. So cells must be a key component of living things. And we'll discuss that shortly. The second leg of the stool is another easy one. Let's put it as this. So all existing cells on the planet today have ancestors, like a mother cell. And it doesn't just apply to us humans, as we have a mother and a father, but it refers to all cells. So a bacteria cell in existence in your gut today will have come from another pre-existing bacterial cell of the same species. So that's an easy one. So cells cannot poof into existence. So that leaves us with the hard one, in my opinion. So that would be the third leg of the stool, right? So here we say existing cells. So this is the third bullet point on this side. So all present-day cells come from existing cells. Then it leaves us with the hard one. And the hard one simply says the fundamental unit of life is a cell. So if you dissect your body, you'll get down into organs, and then further into tissues, and then into cells. But if you continue to dissect the cell, then you will no longer have a living entity, you'll have a sack or half a sack of chemicals. So nothing less than a cell can be living. So the smallest entity that can live is a cell, right? So again, the word cell goes here. At this time, rather than saying that all living things are composed of cells, it says cell is the smallest unit of life, right? Smallest unit of life. So if I told you there was something smaller than a cell, like a piece of DNA, then that would be non-living, because nothing can be smaller than a cell. So there's your cell theory. So what you have to decide is how you're going to remember these 3 facts, and then how will you be tested? So we could tell you, list them all. Or we could say, here's 2, what is the third one? Or, as we'll see in lecture itself, we'll give you a deeper analysis of the cell theory. So that's all you need to remember for this particular concept. So let's move forward. Let's go to the next slide. So the next slide simply talks about unity. And the word unity in the English dictionary means what unifies things, what's common amongst all these things. So we take all the cells on the planet and we look inside them, we find that they are essentially very similar chemically and in terms of their energy needs. So that's what we call the unity of life. Why is that? Well, we believe that this chemical concoction came into existence very early on, about 3.5 to 3.75 billion years ago. So at that time, the basic chemical workings of a cell were already worked out. And since then, things have changed, yes, but not much. So that's what unity means. So the most important way to understand unity is through the concept of the central dogma of life. What is that? That's an observation that can't be violated by living things. So anything that's rigid and is not is unwavering, let's put it that way, is called a dogma. So the central dogma simply says that DNA, as we'll learn a lot about DNA in this course, DNA must first be converted to RNA. And in some circumstances, then that RNA can be converted to protein. It means you can never go directly from DNA to protein in the natural world. Okay, so that's called the central dogma. It's a process. It's a pathway. You start at home, you take your car, you get to your destination. You can't get to your destination directly from your house without taking the car. The next most important thing, and maybe a universal concept in this course, is that information is critical to living things. Without information, critical things, living things will be random, and we can't have randomness in biology. Biology is very, very, very organized chemistry. And any departure from that organization immediately, within a fraction of a second, results in the death of that entity. So a cell will die if it loses its internal integrity. So a lot of energy in terms of food and lots of organization in terms of molecular structure is needed for something to be living. So information is stored in the DNA, and that information may then flow into RNA, and then in many cases flow into proteins, and then those proteins will maintain the integrity of the actual cell. So DNA has another purpose. It is the only molecule that is used by living things to possess information at this level. So RNA cannot be used as an information molecule. And we'll come back to that when we do the exercise in class, when we compare different living entities versus non-living entities. And the final thing is that all living cells must be able to reproduce in order to keep that particular lineage going. If they can't reproduce, then that's the end of that cell line, and there's an extinction will be the next most common outcome. So my cells, they came through my parents, and their cells came from their parents. But apply that not just to human beings, apply this concept to all living things, from fungi, plants, bacteria, and protists. So we'll maintain this theme throughout the entire course. So you mustn't forget that cells have many chemical and energetic needs that are very similar. However, I don't look like a monkey, I don't look like a plant, therefore that leads us to this concept, that we also have to explain the diversity of living things. Why am I different to another species of organism? And the answer there lies in the fact that cells can come in many shapes and sizes. So looking within your own body, your eye cells are different to your brain cells. Your brain cells are different to your neurons. Your neurons are different to your kidney cells. They not only have different shapes, but they can also have different sizes, but more importantly, they have different functions. So a liver cell performs many functions that are different to a kidney cell. Although some functions are the same, such as copying DNA, other functions, like acting as a urine-producing cell, is going to be different to a detoxifying cell in the liver. We'll come into that later. The second bullet point is very important. It says we are all adapted to our environments. That's referring to cells. So a brain cell is adapted to live with other brain cells in your head, and that's the environment in which it has to then operate. But think beyond yourself. Think of a plant. So plant cells have to be adapted to the function that they perform. If you are a cell in the petal of a flower, then you contain a lot of pigment, and that pigment gives rise to the coloration of the flower itself, therefore attracting insects or birds for pollination. So you can see the environment plays a large role in then molding the cells to fit that environment. Different species contain different DNA. So we said to you in the previous slide, all living things are unified by having DNA as their information molecule. What we do know now is that if you look at my DNA and the DNA of a chimpanzee, there'll be differences in that DNA. And those differences are responsible for making chimpanzees different to me. And that is the case for all living things. So comparing DNA, as we'll do near the end of the course, will allow us to understand that there's diversity due to DNA differences. These slight DNA differences between you and me may be enough to change your appearance or your biochemistry compared to mine. So I have black hair, you may have blonde hair, and that can be easily explained by differences in our DNA. And we'll get to that eventually. The last bullet point says that these differences not only make the cell have a different shape or size, but it can change the behavior of the cell. And that's another thing that we mustn't forget in this course, right? We're not looking at just shape and size, we're looking at chemical composition. And that chemical composition can then be altered during the life of a cell to make the cell behave differently. So another thing to consider. That takes us naturally to this evolutionary concept, one of the most powerful concepts in biology. So what it says here is that DNA carries the information, and any change to the DNA has the potential to change the shape or size or behavior of the cell. So mutations are the only thing that can cause DNA to differ between two cells. So mutations, we'll discuss in the future, are permanent changes to the DNA. And they can change the information, or they may change the information. How can you change information? Well, by changing the information of a cell, you can make it different to the parent cell from which it originated. And that can then be either good, bad, or indifferent. So if the change is beneficial, we say that that cell is better suited to its task than the cell from which it came. If it's worse because of the mutation, which is true in many cases, then we say that the cell is worse off than the mother cell or the population of cells from which it arose. And therefore, there's less likelihood that that cell will make it through its life and reproduce itself. But there's also a third category of mutation, and that's where the change in the DNA doesn't really alter the behavior of the cell. And in that case, the cell is no worse off than the parent or no better off than the parent cell from which it came. So there's three outcomes for mutations, right? Good, the bad, and the indifferent. So that takes us nicely to put all these topics together. The definition of life. What does it mean? How do we define life? And unfortunately, biologists are still arguing today as to what it means to be alive. Why? Because there's so much variation in living things that it's impossible to come up with a single sentence that encompasses everything that exists. For instance, there are bacteria that live under the ground that are poisoned by oxygen. There are yeast cells that reproduce by just forming a bud on the side of their bodies. So those differences are unfortunately so great that a single statement doesn't work. So what do? Do we give up? No, we come up with a criteria, a checklist that can be then used to define living things for now, until biologists work out into the future what the answer is to that question. So I've come up with my own learning aid called Mr. Gray Plus. If you watch the longer video of this particular lecture, which is online already, you will find that Mr. Gray Plus has 7 components, which I've summarized here. So in order to be classified living, you have to satisfy all 7. If 6 out of 7 is applicable to you, then you are not living. You have to pass all 7 standards. So the first one says metabolism. So how do we define that? Well, metabolism is basically the sum of all chemical reactions. So living things perform chemical reactions, right? So let's put reactions. Some of these reactions will be building more complex chemistry from simple chemistry, like taking water and carbon dioxide and rearranging the atoms to produce sugar. And that's what happens in photosynthetic organisms. You can also have the reverse chemistry, where you take the sugars and you dismantle them back into carbon dioxide and water. And that's what you and I do when we eat food. But regardless, a living cell is constantly metabolizing. Millions and millions of reactions are taking place every second inside that cell. The next one is important, and we already spoke about that. No cell can live forever, so cells must be able to reproduce other cells. Normally, one cell gives rise to 2 cells, right? So that's reproduction. So let's put that down. One cell then splits into 2 cells. And then those 2 cells get larger, and then they may also then perform reproduction themselves at a certain time in the future. The next one is grow. Grow means in terms of size. So you can start with a small cell, just like this one here. That's going to be half the size of this cell. And then over time, that cell will acquire chemistry from the environment and become the same size as its original mother cell. So all cells have to grow, otherwise every cell division, they get smaller and smaller and then disappear. So growth is a very important thing. And that's to do with chemistry being assimilated inside the cell. Respond. And this is one of the most important ones, believe it or not. If a cell doesn't respond to its environment, then it's a dead cell. Think about it. If the pH of the environment changes, that's going to have an impact inside the cell. And if the cell doesn't adjust its chemistry to cope with that changing pH, then that cell will be unable to metabolize properly, and it will die within a short period of time. So responding to the environment is very, very important. Now, the environment can be both external, which is normally the case, or it can be internal to the cell itself. So it can be responding to its own chemical changes internally. And a living cell is a fantastic object that has the potential to respond very quickly to the needs of its biochemistry. So I'll be mentioning respond again and again, especially for the first half of the course, so that we understand that. Evolve. We mentioned evolve on the previous slide in terms of changes in the DNA that can lead to differences between the new cell and the existing cells. So if you continue this process over many generations, you will find that over time, the descendants may be fairly different from the ancestors. So that's what we call evolution. And that's what Charles Darwin was able to figure out. So evolution is a process that applies to populations of cells. The next one is, this is part of the plus sign, right? The plus sign on Mr. Gray, one of the bars. It says that all living things must possess DNA as their primary molecule of information. So if they have RNA or they have no RNA or DNA, then they can't be considered living. And the final thing is, and this goes back to the cell theory, is that all living things must be made of cells. So if you're not made of a cell, then you can't be classified as living. And we'll prove that in class by doing some activities. So Mr. Gray plus has 7 criteria, and all 7 criteria must be valid for something to be considered living. That's the take-home message. Let's just apply that knowledge. So to me personally, understanding scale is very important. And by understanding scale, you can then put yourself in relative position with other objects. So scale means size. Some cells are extremely long. The longest cells that were recorded currently are those of a giant squid. They can be up to 5 meters long. 5 meters, that's about 6 yards. 6 yards times 6 times 3 is 18 feet. So that cell is 18 feet long. But if you think about it, we also had some giant dinosaurs on the planet, Brontosaurus. And from its pelvis to the tip of its tail, it needed a neuron to control the tip of its tail to sense the environment. And those could have been even longer than a giant squid. They could have been up to 10 meters long, about 12 yards. And that's about 36 feet. So cells can be very, very long, but they're normally very fine. They're not that thick. Some cells are tiny overall. And the smallest cell on the planet today belongs to a bacteria called microplasma. And we find these bacteria in the environment. And not only are the cells small, but their chromosomes are small too. So let's turn our attention to humans. Most of your cells are invisible to your eyes. They're too small to be seen by the naked eye. But there is one cell that is easily viewable by the human eye, and that will be the cell from which you came into existence. And that's called the mother's egg cell. The egg cell is about 1 millimeter in diameter. And you can easily see 1 millimeter with a naked eye if you have decent eyesight. But there are other creatures with larger eggs than us. And the largest animal egg that we know of is that of an ostrich. An ostrich egg can be 10 centimeters. And within that, we have a yolk, and then we have the egg white. Technically, that is a single cell. And what happens is once it's fertilized, then it starts dividing without growing again and again until it forms the tissues of an ostrich. And those individual cells will be microscopic, unable to be seen by the naked eye. So you can see different things. And now the ostrich egg, I believe, weighs about 3 to 5 pounds. So that's a huge single cell. But these are exceptions. The average cell of a living creature is so small that it can't be seen. So what are the sizes that we're talking about? We're talking about 10 to, on average, 10 to about 400 micrometers. So this term micrometer is very important. What is a micrometer? Well, a meter is about 3 feet, just over 3 feet, right? So that would be 1 meter. Now, a tenth of a meter is called a decimeter. Yeah, decimeter. So that would be 1/10. So take a tenth of that, and that would be this size here. The next one, you know, what's a hundredth of a meter? Well, that would be a centimeter. So take a tenth of this, and that would be your centimeter, right? There you go. And then you take a tenth of that, and that's your millimeter. So a millimeter is a tenth of a centimeter. And how big is a millimeter? I think when you cut your fingernails, the amount of nail that's left at the beginning, at the very tip, is about 1 or 2 millimeters. So you can easily see that. Yeah? You can see it on my hand here, the very white part. So a micrometer would be a thousandth of a millimeter, a thousandth of a millimeter. So how far can you see? Well, if you look in your main reading assignment, the textbook, there's a nice figure there that shows you different panels of how different scale is related to each other. So the smallest you can see, if you have good eyesight, is a tenth of a millimeter, which will be 100 micrometers. So the human eye, a good human eye, can see 100 micrometers. So that means you can see some of these cells, but there's not many cells that are that big in the human body, right? Most cells are down at about 100 micrometers. So just beyond the resolution of your eye. So that's where we are. So scale is very important, okay? So remember that, please. And then final part for this lecture coming up, we'll have a little exercise, either in lecture or in discussion, that talks about how do we see these microscopic cells. And the invention of microscopes was very important. And microscopes are simply tools to magnify the world around us. And the most important thing when it comes to microscopes is two things: magnification, how much you scale up the object, but more importantly, the resolution. So the resolution is how much detail you can see at that magnification. So an expensive microscope will give you better resolution than a cheap microscope. So that's what resolution means, the ability to tell two different points as being separate. So let me draw that here quickly. So in an expensive microscope, you will see two objects like this, and you'll see some space between them. So that's considered as being good resolution. In a cheap microscope, you may see this. Here, you can't see a gap between the two. So everything appears to be smeared together. So the resolution of this is poor, and this resolution is pretty good. So to me, resolution is just as important as magnification, okay, when it comes to these tools. Light microscopes use light as their media of magnification. So light waves are magnified and then revealed to the eye as detail. And there's many types of light microscopes. Some of them are simple compound microscopes. Others are bright field microscopes that use various tricks to get the light to be split into different wavelengths. You have polarizing microscopes. In fact, there's a couple of pages in your reading assignment from the textbook, the colored pages, which do a really good job of explaining light microscopes and electron microscopes. So please spend a few minutes looking at that. And then lastly, we have electron microscopes. Electron microscopes don't use light as the media of magnification. They actually use electrons, the particles that run around atoms. So those microscopes can go 10 to 100 times to 1,000 times deeper into magnification and produce results. And there's two main types of electron microscopes, the scanning electron microscopes and the transmission electron microscopes. So, so let's type in here. So I've lost my pen. So we have the transmission electron microscope. That's very much like the light microscope, where electrons are passed through the specimen and then scattered and picked up by the detector. And then we have the scanning electron microscope, my favorite, which is simply bouncing electrons from the surface of the object back to a detector, and then the surface of the contours of the object can then be mapped like a mountain range, where you can see the topography, right? So those are beautiful, and your textbook has examples of both, pictures from both. All right, so that's all we need you to know for this first lecture. When you come to lecture, there'll be various exercises on each one of these concepts. just to reinforce what you know. But you must have read the textbook or watched this video and then gone to the other resources that we have for you. I have to stop now because I've gone on longer than I expected. But guess what? It's for the greater good. Thank you.